This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 674, recorded on October 21st, 2020. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hey, everybody. Here it's partly cloudy, and it's 12 degrees Celsius, 52 degrees Fahrenheit.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is 65 Fahrenheit and a bit cloudy here.
And from Austin, Texas, Rich Condit.
Hi there, everybody. It's 76, headed for 87, cloudy. They tell us we're going to get, I don't believe this, the first freeze maybe in a week or so. That's crazy. But I am looking at a week out, and we're looking at highs in the 60s.
Yeah, we have had more warm weather now. It's 18C today, cloudy. It's delaying the leaves from falling, which is good. That way I don't have to clean them up.
Just for your information, we were supposed to have a hard freeze, but I don't think it materialized, Rich. So I think you can be skeptical like you are. And okay. Minnesota had 8 inches of snow yesterday.
Whoa.
Yeah.
Wow. Our guest today is from Chapel Hill, North Carolina. She is in the Department of Epidemiology at the University of North Carolina. Lisa Gralinski, welcome to TWiV.
Hi, everyone. Thanks for having me. It is 70 degrees here, headed towards 80 and bright and sunny outside.
Good for you. That sounds perfect.
What is Ralph?
Yeah, it's great weather.
Ralph always says southern blue skies.
Carolina blue skies.
Carolina,
yeah.
That's
yes.
right. Carolina blue skies. All right. Well, Lisa, we want to talk a bit about your work today, but before we do that, let's get a little bit of your history. And as listeners will soon find out, there's a connection here. But where are you from originally?
Absolutely. I'm from Western Massachusetts originally, a small town in the hill towns. It's gorgeous. Family posted some pictures of the leaves and the outdoors yesterday. I was really jealous.
Is it near Great Barrington by any chance?
Not too close to Great Barrington. And I would like to note that the town of Great Barrington denounced that statement.
Oh, good. That's great. Yeah, we were wondering last Friday whether they would denounce it or not.
Oh, yeah, they did.
You know, it's funny. A reporter emailed Ian Lipkin about it. And Ian said, no. And he said something else. And he said, because Vincent and I agree on this, we should call it the Columbia. What is it? The Great Barrington. What's the Great Barrington Declaration? So it's the Columbia Declaration. Yeah. Every time someone says Western Massachusetts, I think of Val and Dove.
Sure.
You know? Right? Western Mass. So you grew up in Western MA? Went to high school there?
Beautiful area. Went to high school there. My dad's family's all from that area.
Nice.
Then went to Haverford College outside of Philadelphia for undergrad. Loved it. Really strong undergraduate biology program, lots of hands-on research. And then I headed to the University of Michigan for my PhD, where I worked with Dr. Catherine Spindler.
Miss Kathy to you.
I'm sure you call her Kathy now, right?
Absolutely. Yeah. So I did my PhD studying mouse adenovirus type 1 pathogenesis, how it leads to encephalitis, breakdown of the blood-brain barrier. And that's really where I got to dive into my interest in virus-host interactions.
So when you went to Michigan, were you already interested in viruses?
When I interviewed for grad school, I really wasn't sure if I was going to head in the bacteria direction or the virology direction. I definitely knew I was interested in infectious diseases and microbiology as a general category, but which way it was going to go was pretty up in the air, and viruses are what grabbed my attention there, and that's what I've stuck with.
Yeah. So how far back can you remember being interested in science?
Really, even in elementary school, I think I knew I liked science as a topic, exploring the world around me, academic things. But at that point, I didn't understand what career options were available. As a 7 or 8 year old, I knew there were doctors. I knew there were teachers. I didn't really think either of those were going to be a good fit for me. My father was a high school teacher. So that was kind of my image of a teacher. And I was like, I don't think I like students and children enough for that, which was true.
I wonder if I wonder if the pandemic is going to make more kids aware of science and maybe want to go into science as a career. Just thought of that.
I think so, because also ASV is doing this fantastic outreach where they will connect educators with research scientists and, bring a virologist into the classroom. So just efforts like that as well are going to make a big difference in what people are aware of, I think.
Yeah, for sure. I'm sorry, you were going to say something.
When I was in high school, I got the chance to attend the Smith Summer Science Program. So Smith College is in Northampton, Massachusetts, not too far from where I grew up. And there I got to live on campus for a month and take kind of pretend college classes. They definitely weren't real college classes. And that's where I got to learn about bacteria and labs as a way of life and a career, and start thinking about antibiotic resistance, and, played with my first pipettes. And I came back as a sophomore in high school and was like, this is what I'm going to do. I'm going to be a scientist.
So that was that a summer program at Smith?
That was a summer program. Yeah. And it was a fantastic one.
Can you remember what year that was?
I was there.
This is relevant. My daughter went to Smith. Matter of fact, I have a whole family lineage at Smith.
It's a great school. I thought really hard about going there for undergrad, but it was just too close to home. I mean, it's like 25 minutes from where I grew up. So that didn't have the going out and exploring the world part of what the college experience meant to me. But I think I was there either the summer of 94 or the summer of 95.
That was before my daughter.
Someone said that also recently on a TWiV. They wanted to go to college far from home. Want to be
that
close.
was Kuzmikia, I think.
Kuzmikia. Although then she ended up close to home. She said by then she could deal with that, right?
Right.
Yeah, that was funny.
Being far from home is kind of nice for college.
Yeah. And I was a good kind of moderate distance, 6 hours driving, 5.5, 6 hours by train. So it wasn't too difficult to get home for holidays, but I had a new area to explore. There weren't any old patterns to fall into. It was all new and exciting. And my parents couldn't just drop by.
Yeah, horrors if the parents drop by, right? Okay, so we got through Michigan, PhD with Kathy Spindler. And then what'd you do next?
And then I wanted to continue doing virus pathogenesis as a postdoc. And so after, contacting a bunch of people, I ended up settling on Ralph Barrick in the Barrick Lab here at UNC. And I haven't left. The role has changed, but the science is continuing in coronavirus pathogenesis. And I think it's a good place to be right now.
Oh, yeah. So when did you go to Ralph's lab?
Almost exactly 12 years ago. I think my start date for my postdoc was October 25th, 2008, because it was right around a really critical election as well.
God, that's a month after.
That's smack dab between SARS and MERS.
Yes. Yeah. And I definitely had people question, why are you going to study SARS pathogenesis? This was kind of a one-hit wonder. Do you really think anything's coming back? I was like, well, this is maybe true. But, even at that point, Ralph was really good about thinking about the virus in terms of a model for age-dependent susceptibility. What can we learn about models of ARDS and different things? And now we know that emerging coronaviruses are not going away. So we can learn about them as well as age-dependent susceptibility and ARDS and all those other things.
I also want to note that October 20, 2008 is a month after TWiV began. We began in September 2008. Do you know, just to note to the listeners, I did 2 TWiVs at Chapel Hill a number of years ago. I don't know if you were there, Lisa. One with 7 RNA virologists and one with 7
I was there at that one.
DNA virologists. Boy, that was something because my host blossomed a mania and wanted to showcase virology. So I said, we'll do 2, okay. And that was a lot of people to do. Unfortunately, there weren't many people in the audience. That's what I remember distinctly. But the seminar was well attended.
Well, that's because all the virologists were on stage, as it were.
That's right. That's right.
So what started as a postdoc morphed into an assistant professorship. Is that right?
Yeah. So I stayed on as a research associate for a little bit and then transitioned to a research professor position. And now kind of the pandemic has provided an opportunity for those of us who are in the coronavirus field. And so myself and one other person are transitioning to the tenure track, which is a really exciting opportunity. Kind of weird to be grateful for the pandemic, but I guess we'll take some good with all the bad.
Well, congratulations.
Thank you.
So I think your tagline on Twitter is you're in the Department of Epidemiology, but you're not an epidemiologist, right?
No, I definitely don't have any epidemiology background. I've done a lot of reading this year. It's in the School of Public Health. We make a lot of sense there, but it can be really tricky sometimes when people reach out for epidemiology commentary or they want advice on a model. And I have to say, oh, that's not my expertise. If you want to talk about how the virus replicates, how it enters cells, how it interacts with host proteins, we can talk about that, but I should not be doing modeling.
Yeah, I know for years Ralph has always been part of the Department of Epidemiology, but I think this year, the first time we had him on, we asked him an epi question. He said, that sounds like epidemiology. I don't want to answer that.
We have great people in the department who do that. I think we're working on a lot of collaborations, but...
So Lisa, I going through PubMed, your publications, I count you on 13 SARS-CoV-2 papers. And you've also worked on SARS and MERS. So you've covered them all over the years. And one thing that strikes me is these are team efforts, right? There are a lot of people, and I'm just wondering how that works. How do you figure out who's part of the team and who does what and so forth? Because that's never obvious to us how that works. And I've never been part of a huge effort myself. So I wonder if you could shed some light on that.
The first thing I would say is thank God for all the people to share the work this year, because it is far too much for any one or two or three people. So we've always had a really good team-minded approach, in the Barrick Lab, coming in as a postdoc and now trying to start my own independent projects, but still contribute to things there. So with mouse work in particular, that's pretty easy, where you have mice that are going 7 days, 10 days, and they need to be weighed every day. They might need to have some other specialized measurements taken. So people come in, help take a day or two, maybe give you a weekend day back to yourself. And so you reward them with authorship. But when we get into more specialized assays as well, or now when we're taking a newly generated recombinant virus that somebody has made, and then we're putting it into animals as quickly as possible, and we're trying to do kind of 12 things at once all the time, being able to share the burden is incredibly important. And the other thing is we're all excited about this work. We all want the opportunity to be on these first wave SARS-2 papers. And so this way, nobody has hurt feelings. Hopefully nobody's burning out. And we can respond as quickly as possible and try and make a difference.
Yeah, one thing that's remarkable is the speed at which these papers, not just from your lab, but from many others, but you have published some amazing pieces of work. And I think you're right. It's only because there's so many people involved, right?
Yeah, 13 SARS-CoV-2 papers, and it's been how many months?
It feels like decades, but it's only been about 10 months.
So are there gigantic lab meetings, or are there subgroups? How do you keep up with and process the information?
So we used to have more subgroup meetings where we would have a bigger coronavirus group meeting, and then there are actually flavivirus and norovirus arms of the lab as well, although they've all kind of been swallowed into the coronavirus group right now. So kind of earlier in the pandemic, lab meeting was just canceled because we were going crazy trying to keep up with everything. And then kind of early summer, we were able to restart that again. I used to actually organize lab meeting, but someone who's in the flavivirus group has taken that over, which is a huge thank you, Ellen, for that, because I would never get around to sending the emails out with everything else that's going on. And so kind of we do general lab business, which is kind of okay, guys, this is what we're running low on in terms of PPE. Be careful on that. We need to watch an ordering deadline coming up, kind of general maintenance issues, because we're not all seeing each other face to face the same way anymore. And then kind of one person will present. And luckily, we've had lots of chances to celebrate papers and good work that people are doing. So that's encouraging. We also sometimes just have it on Zoom in the background while someone's in the BL3, and there's no camera, no microphone there, but they can at least listen and run over to the chat if necessary. This year's really all about multitasking.
So there must be sort of lead scientists on the various projects that coordinate the effort. Is that right?
Yes and no. We're working more towards that, but traditionally, even though we've had people of all different levels, we've operated as a very flat structure. But now we're bringing so many new people in, and there are so many projects going on, and many of them Ralph can't be a part of. So Tim or Rachel or myself or someone else will run that project as PI. So now we have a little bit more of a structured hierarchy going on.
And in terms of actually writing the paper?
Massive group effort.
Okay.
So I've heard different labs having difficulty this year in terms of things like being able to order supplies and being able to, get people working with distancing requirements and things like that. How does that work in the labs that are working on SARS-CoV-2, where you're trying to get everything done so rapidly?
We are pretty much all working 150% of the time, I would say. We're not really doing the 50% people on site. We have expanded into several conference rooms on our floor. The university has opened those up for us so that we can spread out a bit more. In mornings when I'm doing emails and paperwork and more business things, I'll try and stay home and do that. But a lot of it has to be on site in person, and to respond as quickly as we need to. I see a lot of my coworkers face to face every single day. If we're in the BL3, it's fine. It's probably the safest place to be this year. And it's really nice to not have to be wearing a mask in there, honestly, and just be able to see people face to face like normal, not over a computer screen, not behind a mask, not worrying about being too close to each other. So that's kind of a strange aspect. That's a bonus of being a coronavirus scientist this year.
For the listeners in the BL3, not having a mask is because you're wearing a big hood, right? And
absolutely.
Right.
Yes, we're wearing PAPRs. We have massive amounts of TWiV gear on. So we have our own independent respirators. So we're feeling very safe in there.
So when did you, I wrote in the notes, when did you go, uh-oh? I think you know what I mean by that.
Yeah. So it's funny thinking back on this. My husband and I love to travel, and we were in Guatemala and Belize over New Year's. And so I remember it was New Year's Eve. We had an early start the next morning because we were going to go see Tikal, actually. And I'm scrolling through Twitter, trying to fall asleep, but there's a lot of noise outside because it's New Year's Eve. And I think it might have been Helen Branswell retweeted something about pneumonia cases in China, a cluster associated with the wet market. And I kind of turned to my husband and was like, that's really interesting. That's the type of thing that could be a coronavirus. And so I would just kind of pay attention every couple nights as I would be online before bed as we traveled. And then I got back to the US kind of first week in January, and said everyone was super excited about it. Lots of emails going around, but there was this lull where we were waiting to find out, is it dying down, or are we just not getting information out of China? And there was actually a conversation between a couple of us in Ralph's office where he said, wouldn't it be just like a coronavirus to cause all of this fuss and then turn out to be nothing? And, kind of mid-January, that statement made sense. And now that we're here late October, that's very clearly not what happened.
When did you start, full on working on the virus?
So the cloning side of the lab started as soon as the sequence was available. Okay. I think it was January 10th it came out. I'm not one of the expert cloners in lab. I can do that, but it would be a much slower process if I was leading the effort to generate the infectious clone.
Yeah, man, there are some people who can just do this. Yeah. It's amazing.
Yeah. So Jacob and Kenny and Boyd and others were kind of full tilt on from that point. And then it actually got sped up when we had an isolate from the Washington patient here in the US. And, we got sample from the CDC, and then we were able to get RNA from cells infected with that isolate. And so that sped the process along. We weren't waiting on plasmids coming in from BioBasic anymore.
And it seems like I remember that you were about to have some mice go to Jack's to be cryopreserved, and you pulled that back. So you might have been involved with that.
Absolutely. Our lab had human ACE2 transgenic mice that had been sitting around largely unused for like years and years and years. And so we were finally going to cull the colony and send off for cryopreservation. And we were down to maybe 6 cages with just a couple of mice each. And, we went to that first lab meeting back in January, back when we were in person, and it had just been declared that it was a coronavirus. And so at lab meeting, I emailed our mouse colony manager, Kendra, was like, Kendra, do not cull any more of those mice. In fact, put all of them together to breed immediately. So we had some mice available, but honestly, they were older. They didn't breed that well. So we still needed a couple generations to get enough mice to be able to do anything with. And by that time, we were actually pretty close to having a mouse adapted virus that we had generated in lab. So we've used the transgenic mice a little bit, but actually not that much.
So I wanted to go back a bit to a paper which you're part of in 2015, and which we did here on TWiV a long time ago. A SARS-like cluster of circulating bat coronaviruses shows potential for human emergence. And I think we had Vineet and Ralph together on that episode. I don't remember. Anyway.
I think you did.
So remind us what that's about.
So that paper took sequence data from Professor Shi's group at Wuhan Institute, where they had done surveys of bats in caves, I think in Yunnan province and other areas in China, and had these full-length sequences of coronaviruses that looked very similar to the original SARS from 2002, 2003. And from that work, it wasn't known if these viruses would be able to infect human cells at all. And so that was really where we picked up. Initially, Vineet made a chimeric virus that expressed the spike protein from these bat viruses and put them in the SARS backbone. And honestly, our prediction was that they would not grow, that they wouldn't work at all. And we were shocked to find that they did, which shows you that modeling is not always correct. And that's why it's important to do the wet science.
So why did you think that wouldn't work?
Looking at the residues that would come in contact with ACE2, it didn't seem like they would be able to bind. It seemed like there was going to be a receptor mismatch.
Okay, so just based on the receptor. Okay.
But lo and behold, they could replicate in human cells, which was startling, but meant we had something really interesting going on. And so we did a lot of characterization with the chimeric virus, and then also generated the full-length virus, which took quite a bit longer, and showed that these can infect human primary cells, that they can infect mice. SHC014 can cause some disease in mice, a little bit of inflammation in the lungs, cause some weight loss, some changes in respiratory function, and that it's not so surprising after MERS came out. There was SARS and then there was MERS, and you have one coronavirus that can emerge and cause a lot of disease in people, could be a fluke. A second one comes. Probably at this point, there are going to be more in the future. And the data with SHC and then the WIV1 follow-up paper in PNAS showed that, these viruses are out there that have potential to infect people. And now we're in 2020, and it's abundantly clear that this has happened again.
Okay, so just so that I've absolutely got this straight, you had sequence information on SARS coronaviruses from bats. A lot of them looked like bats, viruses that had spilled over. Selected a couple of these, cloned them out, so now you got virus, and showed that they'll infect appropriate human cells in culture, right?
Yes.
Wow.
Yes. Very genetically distinct from the current SARS-CoV-2, but related.
Yeah.
So if that's not a warning sign, nothing is.
We thought that was a really big warning sign. It was controversial and high impact at the time, especially since there were, gain of function and pathogenic pandemic potential virus issues happening at the same time. But then following that, it was actually a letter from Professor Xi's lab that came out in 2018. And Ralph referred to this for quite a while. It was very difficult to finally find on PubMed. I think it's like China Sinologica or something. And they went back to the community surrounding these bat caves in Yunnan province, China, and took blood samples from villagers in the area and showed that about 3% of the local population had antibodies in their blood that could detect the nucleocapsid protein of these bat SARS-like coronaviruses, and that people in Wuhan province did not have any of these antibodies. So saying that there does seem to be some low-level exposure to these other coronaviruses. So I think those two pieces of data together really powerfully showed that there's a lot of potential here.
Yeah, in fact, you point that out in the abstract. You say, our work suggests a potential risk of SARS-CoV reemergence from viruses currently circulating in bats. So, obviously we did nothing to be prepared for SARS-CoV-2, but looking back, and I'm sure this bugs you on a daily basis, what could have been done from that? If we had resources, if we had unlimited resources, what could have been done with this information, do you think?
I think something more along the flu surveillance model lines, where we constantly hear about groups that are out looking at new flu strains in Southeast Asia and other hotspot areas, where you're looking in people, you're looking in suspect animal populations. So coronaviruses, that would be bats. And to say, what do we see that's kind of percolating along at a low level? Do we see something that is, on a sequence level more similar to what we're really concerned with? And are there other sequences that are really a big question mark, and we need to take them back into the laboratory and do a hands-on experiment kind of in the model of that 2015 paper?
So more surveillance more surveillance than was done, right?
I'd be interested in your perspective on what actually happened in this case. Now, this may be almost all speculation, but the phylogenetic data comes to the conclusion that SARS-CoV-2 may have arisen in bats as much as decades ago, but it's not clear, really, at least to me, when it actually spilled over into humans, whether there was further adaptation in bats or humans. Do you have a speculation or an idea of what actually happened?
I don't really have a favorite hypothesis on that yet. I read different papers, and there's lots of talk about pangolins, and yes, there's a furin cleavage site in the spike there, but the rest of it doesn't make that much sense. The virus has clearly evolved in some species at some point to be very good at transmitting from person to person while retaining a lot of pathogenic ability. And it's hit this terrible sweet spot that other human coronaviruses haven't, where it spreads really well and it causes enough disease that we really need to be concerned about it. And the other ones have one of those abilities or the other. And a lot of questions remain about, is there a reservoir species between bats and people? That's something I'm really interested in. Getting back into China to do really extensive, exhaustive searches there, I think is going to be very difficult. We have partnerships with Peter Daszak's group and the EcoHealth Alliance as part of the new CREED network, where we'll be looking at other places in Southeast Asia, but we won't be in mainland China. I think we'll be in Vietnam and Malaysia and other places.
And Peter, who came on TWiV a couple of times, has said that those are also important places to look at because there's a lot of potential, right?
Absolutely. I think especially Vietnam, they found some really interesting sequences.
So what's the barrier to going back to mainland China? Does that come from the US or the Chinese or both?
My guess is both, but I'm not really sure which is more prominent on any given day of the week.
Okay.
That could change,
so one
right?
thing I've noticed.
I'm sorry.
One thing I've noticed
Go ahead.
That really surprised me is I pulled up this paper from the Nature website as we've been talking, and I don't know if anyone else has noticed this. This is the first I've seen it. There's actually an editor's note at the top that says, we're aware this paper is being used as a basis for unverified theories. There is no evidence that any of this is true. Scientists believe that an animal was the most likely source. And I'm sort of like, go nature for getting into this.
Yeah, that paper, which we thought was like this amazing thing saying, hey guys, you need to watch out for this. And then it actually happened, has definitely been used by a lot of groups to start all sorts of conspiracy theories. I'm not going to point people at those areas of the internet, but it's made life really complicated for all of those of us who were an author on this paper, Vineet and Ralph especially, but anyone who's on the author line. And I'm just surprised, 2020, folks.
I didn't know that Nature would get in the middle of it, and I'm glad that they have this note.
That's good. Well, we've spent a lot of time on TWiV defending all that work, and back when it happened, there were the gain of function people who criticized it, and we told them they were wrong. And then they came back and said, hey, look, they were doing this, and this is why we have this virus now. So it's just crazy. We've tried to do our part here.
We appreciate it.
Another paper I wanted to touch on is also from 2015, Genomewide identification of SARS-CoV. Again, it's the first SARS susceptibility loci using the collaborative cross. And this is, you're the first author on this one. And so I'm guessing that pathogenesis is your forte, right?
That's my area of interest for sure. That's where I spend my time.
Okay. So tell us what this was about and what you learned.
So this paper was taking advantage of this genetic reference population that is largely housed at UNC. So we have particularly easy access to it, but it's also available globally through JAX, through, I think, the Pasteur Institute, investigators in Australia. So the CC was designed from 8 standard inbred lines to take in, I think, about 90% of the genetic diversity of the mus musculus genome. And we have these recombinant inbred lines, which are now reproducible, genetically unique, and have lots of genetic diversity. And the level of SNPs and indels is pretty reflective of what's seen in the human population. So that's the rationale behind the CC. And we had early access to that here at UNC using mice that weren't yet fully inbred. And as kind of a proof of concept study, our group infected these singleton mice with SARS-CoV, mouse-adapted SARS-CoV. Mark Heisey's lab worked with influenza. Other people studied other aspects of the immune response and growth and all sorts of different things. And so we essentially performed a mouse GWAS study to look for susceptibility loci, regions of the genome that were associated with different markers of SARS pathogenesis. So, weight loss, virus titer in the lung, different histology phenotypes, that type of thing. And we followed up on one phenotype in particular, which was vascular cuffing. So recruitment and accumulation of lots of different inflammatory cells around the vasculature in the lungs of these mice with SARS-CoV. And really the tricky part of all of these GWAS studies is the validation and the narrowing in on one gene and showing that it has the specific function that you predicted. And we were really lucky in the case of TRIM55 and this chromosome 3 QTL, where there ended up being one gene, one pseudogene, and one non-coding RNA under our locus of interest after we did a lot of genetic and statistical kind of tricks to narrow things down. And the one gene ended up having the phenotype that we predicted. And so it was a very exciting day in lab.
Go ahead.
The vascular cuffing, this is a pathogenesis phenotype that shows up in the lungs of the infected mice.
Yes.
Is that right?
Yes. You see it some in humans. You see it on all sorts of histology.
Well, that's where my question is going, is whether, my general question about using the collaborative cross is that you're going to infect all these mice and you're going to come up with, say, respiratory symptoms of some sort or another. But do those respiratory symptoms, when you get down to the histology and get down to the molecular mechanisms, are they really the same as what you're seeing in SARS infections in humans? For that matter, is SARS-1 the same as SARS-2? Or are there differences? Because I'm getting a sense that there are many different ways to skin the respiratory disease cat.
Absolutely. And a lot of it is just going in with previous knowledge from SARS and MERS. But I'm definitely going to say, like, watch this space for future publications on QTLs and maybe broad screens of CC mice, and maybe taking that back to recent human GWAS studies with SARS-2 patients. I think there might be some really cool data coming.
Yeah. Okay. Yeah. We looked at that paper very recently, the one from the guy at Rockefeller, whose name is escaping me, the French guy, right?
Oh, Jean-Laurent Casanova.
Yeah, that's one of them. Yeah. So the gene you identified in this paper, was there any evidence that in humans this plays any role?
Not so much in this case. Vascular cuffing is a little bit tricky of a phenotype where it's definitely observed following infection, but it doesn't have a strict association with pathogenesis. So we don't know that it's like critical for disease. And TRIM55 is a really unstudied member of the TRIM superfamily of proteins. So this one ended up being a little bit more on the basic research side, but some of the stuff that we have coming definitely translates really well to people.
Just the experience of doing experiments in the collaborative cross. You're going to learn something about pathogenesis that's applicable somewhere, but it seems to me also that validation of the model and just learning how to deal with the mice is important.
That was a lot of the goal of this, was the proof of concept to say, we've taken it down to a single gene once. You can trust us to do it again with more resources and more people to really dive into all of the nitty gritty.
So is the collaborative cross still evolving, generating new inbred lines and et cetera?
So they're not starting any new inbred lines, but some of the ones that kind of made it through the bottleneck of inbreeding are not super robust in their breeding. So they're constantly trying to improve colony maintenance and that type of thing. So lines sometimes go in and out of availability if you're looking at the Systems Genetics Core website or that type of thing.
I'd like to make an aside here, and that is that Lisa and I have discussed the fact that when she was in my lab, she did not work on the project that we had in genetics of susceptibility, where we were doing positional cloning to identify genes involved in susceptibility of mice to mouse adenovirus, and how once she got to her postdoc, that all changed. So she learned some things, I think, by osmosis in terms of the genetics being in my lab, but really went into the deep end when she started the collaborative cross.
Well,
Absolutely. It was really funny that that project was not really on my radar as something to be a part of in grad school at all, but it was always right there hearing about the progress, day to day, and really cool results. But I didn't want anything to do with it because it was too much genetics. And then I got to UNC and wow, there are a lot of genetics involved in my projects now.
So how many different lines are there now? I don't know if you call them lines or strains or whatever, but inbred lines, how many different mice are we talking about that I have to infect to set up an experiment with a collaborative cross?
I think there are about 70 that are available, but in our experience from like doing screens for new phenotypes, if you get in 10 pretty random lines, if you just ask for lines that breed well, aren't terrible to handle, and have pretty good genetic spread, you will probably see a phenotype in whatever it is that you're interested in. So we've done this with, I've screened CC lines for susceptibility to SHCO14, and 9 out of 10, their weight loss curve just stays right at 100%. And then that one line, will lose 15%. And you say, okay, that's the one I'm interested in. Maybe this is what I want to start an F2 with, or I want to dive deeper into this as a new model.
So I'm trying to, in my mind, once again, for the listeners, sort of describe the rationale for this whole thing. As we've discussed many times before on TWiV, it can be difficult to find an appropriate animal model for a disease. The standard inbred lines of mice might not show a disease with a particular virus. And so you could go and gather mice or whatever from the wild or other critters from the wild and infect them and maybe get a disease, but you don't know much about their genetic background. Now, this is an opportunity. I'm making this up. Just tell me if I got it wrong. Okay.
It all sounds right so far.
This is an opportunity to take a bunch of different, basically strains, if you like, of mice, okay, and look and see if you can reproduce an appropriate phenotype in one of those. But the real advantage is that you know what their genetics are. So when you do
Absolutely.
see susceptibility, now you can trace it back and get some more immediately, get some real handle on why it is that they're susceptible relative to these other guys, right?
Absolutely. Like if you buy the CC recombinant inbred mice from UNC or JAX or wherever, their genomes are already known. You don't have to pay for the genotyping. You don't have to do the alignment. And they're reproducible. The stock is surveyed, it's maintained at a high quality. So you can come back to this resource over and over again.
I should say that Ralph's lab was kind enough to send us 8 CC lines for our project, and that's all you need. I can tell you.
I hope they weren't the terrible little jumpy ones.
One of them is very jumpy, yes. And I think one doesn't breed as well as the others, but they're all working out well. Yeah.
I picked out this incredible phrase from, I don't know, the discussion or something like that. Just quote, this is some sort of concept. I think I kind of get it. Transgressive segregation or release of cryptic genetic variation. And I feel like, whoa, I'm on another planet now.
Same. That's where I double check with the genetics collaborators. Like, can you just make sure this makes sense? Can you maybe flesh out the details a little bit? But in a nutshell, what that is getting at and what is really cool is that if you take 2 lines that have pretty disparate phenotypes and you cross them together, what we see in the CC is that the phenotypes of the offspring exceed, go past on either end, the phenotypes in the parents. And we saw that even in that 2015 paper where I think we show the weight loss and titer phenotypes for the 8 founder strains and then each individual pre-CC mouse. And you can see mice that have like screaming high titers in the lung and mice that have practically no virus in the lung. And then the 8 founders scattered throughout that range.
This is Mendel on steroids.
Yep. All right. I wanted to ask you about another paper. These are still pre-SARS-CoV-2, but I think that's important, right? And we learn a lot. This was 2018. Combination attenuation offers strategy for live attenuated coronavirus vaccines. And this is obviously relevant to the vaccine effort. So what did you do there?
Absolutely. So this was live attenuated viruses, which aren't really being pursued at this point for SARS-2, but are potentially an option. And you tend to end up with, really robust T cell immunity and a stronger response than you see with like a vectored vaccine or a subunit vaccine, or the mRNA vaccines are obviously very new. So in this case, we took advantage of what we knew about the coronavirus genome, how it replicates, how the virus hides for the host immune response, and engineered 2 mutations, which on their own are both somewhat attenuating, and putting them together, we kind of have a safer attenuated virus. So in this case, there was a mutation in NSP16, which is part of the replicase complex, has methyltransferase function where it helps hide the virus RNA from the host. And so when this NSP16 mutation has happened, you get a much stronger interferon response, and virus replication is clamped down on much more quickly. And then also a mutation in the NSP14 gene, which is the exoribonuclease, which is the proofreading activity, which is part of why coronaviruses can have such a large, stable genome. And so if you mutate that domain, you have kind of a mutator phenotype with your virus, and it doesn't replicate as well and is also attenuated for pathogenesis. So by combining these 2 mutations in one, we had a virus that even when it was put into RAG knockout mice, so mice that are massively immunocompromised and they don't clear virus, so you can infect them and 30, 60 days later, you have pretty much your input virus still sitting there. Somehow they don't get sick, but they don't clear. So we use those as a model of kind of an immunocompromised person. If you were to give an immunocompromised person this vaccine, would it have a chance to revert back to a virulent model? Because that's always the risk with a live attenuated vaccine. So by putting in 2 mutations, now you have, I think, like 5 or so residues changed in the genome. The virus is unable to revert as it was with just one of those mutations. And it's potentially a safe live attenuated vaccine.
And this, you also immunized mice and you challenge them and show that they're protected from disease, right? So
no weight loss, even in old mice, which are hugely sensitive to SARS-CoV infection.
So the question is then, you said earlier, no, this is not being pursued for SARS-CoV-2. Why is that? Do you know what the reason is?
Probably because the safety testing on something like that is looking at a timeline that's much longer than we really want for these first wave vaccines. If the pandemic really lingers, I can see that companies might become more interested in this. But right now we're still at the point of making these types of mutations in our infectious clone system and starting to do similar tests. So just the timing is a big part of it. Making a subunit vaccine is much more straightforward. Sure. We've all seen the mRNA vaccines like race to the front of the list just because they can produce so much so rapidly.
Yeah. I heard that.
Do you think there are any positives or pros to having a live attenuated like this one?
I think they can elicit a really robust immune response. I've always personally been a little sketchy about putting something like suggesting a vaccine that one of the mutations drives a mutator phenotype. To me, that seems like a massive regulatory hurdle. Like, how could you ever guarantee that this would work? But, 60 days in a rag mouse with no mutations is a strong suggestion that there wouldn't be an issue.
So the methyltransferase mutant, if I understand this correctly, the attenuation conferred by the methyltransferase mutations was okay. The problem was the reversion. Is that right?
Yes.
So could you not make a deletion of that gene?
So it's a replicase gene and you can't get rid of any of those entirely. What we can do is get rid of certain catalytic residues and lose, some functions of the gene, but we need the rest of it to be there.
Why? Does it have another job, or is its role as like a structural component in the replicase important?
This is where we reveal that I'm a pathogenesis person and I'm not actually great at telling you all of the functions and like the multiple functions of each structural protein. It definitely binds with NSP10 and has a lot of roles there, but I don't remember what they are.
So it's quite possible, here I'm making this up. I don't know squat, but I have dealt with pox viruses have a methyltransferase that does the same thing that I've spent time on.
Is it for capping? Methylating cap rich?
In the case of, and it does the same thing. It helps the virus to hide from the innate immune response. And in the case of pox viruses, that function is on the same protein as is important in polyadenylation, and also has an elongation function. And I've always suspected that same protein is probably part of a complex. Okay, so that if you get rid of it completely, you might not only knock out some other functions, but destabilize the complex and compromise a lot of different things. So that could be it. Now, likewise, the exonuclease, I've always thought that those mutants in the proofreading function, it would be hard to grow stocks to start with, and in particular, maintain stocks that you were confident had the appropriate genetic background, because I would think it would be going bonkers all the time.
Yeah, so that's a big advantage of working from infectious clone systems. It's a little bit slower, but we never work beyond a P2 virus. Okay. So we don't have as many generations of replication to cause all sorts of problems. And especially with those XON viruses, as we call them in lab, the NSP14 mutants, they do grow more poorly. So you end up having like a log or two growth reduction in cells. And then in mice, it's multiple logs. They don't do much at all.
Kathy, I noticed she says infectious clones, very well trained. Mm-hmm. I don't want to say the other word. By the way, did you say, is an inside TWiV joke. Did you say, Lisa, that you're doing this for SARS-CoV-2 and you're going to see how they work in mouse models?
Yeah. So We have been working on recovering an NSP14 mutant virus along with Mark Dennison's lab. We've had a long-standing partnership with their group working on NSP14 and XON mutations. That's been really fruitful. The virus is being a little challenging to recover is my understanding. So I'm not sure either group has it yet, but it's also one of those situations where there are so many other things going on that I don't think anyone is really just strictly focusing on trying to get that virus to recover at this point.
You said, by the way, if this pandemic lingers. So what do you think? Is it going to linger?
Well, it's certainly not going anywhere right now. I think a lot of it depends on these early vaccines and how well they do for protection, how willing people are to take them, how good our production lines are, distribution. You know, thinking about where I am now, working at a major medical center, they could handle like a Moderna or a Pfizer mRNA vaccine and those storage requirements. Thinking about where I grew up in Western Massachusetts, absolutely not. I'm in a town of 2,000 people with a Hilltown Health Center that covers the surrounding, 8 or 10 towns. They don't have a -80 freezer. They don't have enough people to give all of those doses together that come in one pack. It's complicated.
Yeah. They're all important points that we kind of haven't thought about. We're just thinking about the vaccines, but yeah, that's going to be an issue.
I have visions of when the first polio vaccines came out, and we talked about this before. There were immunization programs and I lived in a fairly small town, and we all gathered on an appointed time in the high school gym and lined up and got immunized.
And they used glass syringes with needles that had to be sterilized and often just boiled because there weren't autoclaves. If you say no -80 freezers, they didn't have autoclaves everywhere back then. I remember Jonas Salk's son told me they got the first vaccine in their kitchen, and the father boiled the syringes on the stove.
Wow.
I have a friend who's a hospital pharmacist in El Paso, which is a good sized town. But he said that buying 1 or 2 -80s for their hospital pharmacy is no small deal. It's money, and there's going to be a run on -80 freezers. Those don't grow on trees.
Yep. All right.
And it's something that we don't always think of, I think, as virologists. Our lab is full of -80s. And so realizing that smaller doctors' offices, medical centers don't have this type of thing, like, of course they don't. They don't have the need to have frozen cells available and have viruses available, but it's going to be a challenge.
All right. One more paper I wanted to ask you about. This is a SARS-CoV-2. This is a paper where you look at a drug called EIDD-2801. And, we're not hearing too much about antivirals these days. The ones that were repurposed, some of them didn't work, and remdesivir is okay, but it's not the magic bullet. So what's happening with EIDD-2801?
It is in phase 2 trials actually right now. UNC is a site. And what's really, really, I think, a big advantage about this drug is it's delivered orally. So the complicated part with remdesivir is that it's given intravenously. So it's an inpatient hospitalized setting. So I think almost everyone who's getting remdesivir is probably getting it too late. But with EIDD-2801, patients can come in as an outpatient, get some pills, take them home. And so you're going to be able to reach a much larger number of people, hopefully get them earlier in disease while you're still really in a virus replication phase and not in a immune response causing all sorts of havoc in your body phase. And fingers crossed that it works as well in people as it works in mice, because in mice, it works fantastically.
Is it an RNA polymerase inhibitor?
No, this one is another nucleoside analog.
Nucleoside analog. Okay. Similar to remdes. Okay.
So in this paper, you use something I've never heard of before, which are lung-only mice.
Oh, yes, right.
I had never heard of them before March also.
So, can you tell us a little bit about them?
So these are non-skid mice that have had fetal human lung tissues, tiny implants put under the skin in their back, and then it's allowed to grow for a number of weeks. And you end up with this organoid that has something like 30 cell types that are all reflective of what you see in human lungs. So you have your type 2 pneumocytes, you have alveolar walls, all of these structures. They look a little bit different because they don't have air inside of them. So that is very kind of weird for me to see still when I'm looking at slides. And so the lung-only mice, there's no human immune components that have also been, instilled into these mice. We're doing this work with Victor Garcia's lab, working with him and Angela Wall here at UNC. And so they also have BLTL mice, where it's these same mice with the human lung implant, but they are irradiated. They get hematopoietic cells from humans put in, so they have some human immune response there. And then they also have a thymus liver kind of tissue sandwich put in, attached to the mouse spleen as well. So dissecting them is kind of weird.
So did you say sandwich because they're BLT mice?
They also literally make a sandwich of the tissue, but yeah, it's kind of terrible, but
yeah, that dissection does sound odd.
Yeah, I wasn't really prepared for it the first time, and then I was like, "Oh, okay, yes, this all makes sense."
So in the lung-only mice, if that's what you call them, you said they're a non-skid background, so that's severely immunocompromised. You do that so they won't reject the foreign tissue, right?
Yes.
But at the same time, that means that they're not going to display, in response to the infection, an appropriate, necessarily a relevant immune response. What you're going to learn mostly is what cells and what structures the virus infects. Is that right?
Absolutely. We kind of don't have the complication of an adaptive immune response coming in. There's some innate response from the local tissue that's actually infected. And because there's the receptor incompatibility with SARS-2 and mouse cells, we don't have to worry about a mouse response or any mouse cells getting infected either.
Can you tell a little bit about how you actually infect the lung-only mice then?
Oh, it's interesting. Angela shaves the lumps on the backs of the mice before she brings them over to me. Then they get transferred into our satellite facility. We anesthetize the mice just like normal. And then you actually have a syringe, like a needle with the virus, and you inject straight into the organoid lump.
Yeah, because
so
these
direct
are
access to the human tissue.
These are not actually exchanging air or anything. They're just collections of cells. Yeah. Interesting. So you said this is in phase 2 in people, so it had a good phase 1 safety profile, I guess. Right?
Yeah. So this drug has been around for a while, and there have been some concerns about safety and like mutagen effects, but it passed all the safety trials in people with no problem. I think it probably also helps that it's not a drug that you're going to be taking for a long, long time. You probably wouldn't want to be on this for months, but you're not going to need that, hopefully, to treat your SARS-2 infection.
How interesting that we don't hear much about antivirals like this. We hear about remdesivir and monoclonals and vaccines, of course, but ones like this, we don't hear much at all.
Yeah. Well, hopefully it gets to phase 3 and then maybe we'll hear more.
Yeah.
There's a wiki page on this, and maybe you already said this, but apparently it was developed for flu initially. Yeah,
it has a long history. It's been tried for a number of viruses. I was really surprised going back to the publication history. And so EIDD 2801 is the prodrug form. So you might also see it reported as NHC or EIDD 1931, kind of all coming down to the same thing coming out of the Emory Institute of Drug Development.
It's a pretty simple little dude. It's a pretty straightforward nucleoside analog. And I would imagine the prodrug gets processed, metabolized to a triphosphate form, in which case looks to me like just hydroxylated, an amino hydroxylated form of cytosine. Pretty straightforward.
Yeah, it gets incorporated as the cytidine, and then, which is pretty okay. But then when it comes to replicate, sometimes you get a U put in instead. And so then you start running into.
So it's mutagenic.
Yeah.
All the same as what? Ribavirin.
So one thing that I learned from Mark Dennison, that not every nucleoside analog will be inhibitory because of the exonuclease, which can correct a lot of the errors that are put in. So he's like, remdesivir works, this one works, but not every one will work. So you have to screen more drugs to find the ones that work. All right. Anything else from anyone before we say goodbye? Kathy, is this the Lisa that left your lab, or has she changed at all?
She's changed a lot. She's learned a lot, and she's become quite the expert.
Good.
Thank you. I'm still not a morning person, though. I will say that.
So I guess the one question I have is whether, you have 13 SARS-CoV-2 papers, and we've really, we haven't been able to talk about all of those. So is there anything that we should have talked about or that you find really exciting and interesting that you want to make sure we mention?
I think a lot of our work so far has been using platforms that we developed to, establish models or test different drugs, different vaccines, different monoclonal antibodies. And I think what we're all really excited about moving forward is being able to do more of our own really basic research and still be a community resource and do our part in the pandemic response, but get down to some basic science about, what is unique about SARS-2 versus SARS-1? What can we learn about different aspects of the host response? We're looking at coagulation. We're looking at immune modulatory drugs, kind of all of the things going forward. So many different aspects that we're really excited to be jumping into.
That reminded me, I wanted to ask about the work that you got funding for that was submitted before SARS-CoV-2 even came around. Are you going to be able to get back to that in some way? Your so-called day job or whatever.
That's definitely been a struggle. If we could write out our 80-hour weeks instead of 40-hour weeks about what we're working on, a lot of that has paused, but is now starting to pick back up. The R21 that I was funded on earlier this year is looking at wild type coronavirus infection and other susceptibility alleles in the CC. And so, we've had some F2 mice breeding in the background, and I really need to start infecting them. Because age is important in phenotype. So there's a narrow window of opportunity. I think, NIH has been pretty understanding that we needed to divert efforts for a while. And the lab is also growing to help with new projects and keep up with old projects. But we need to get back to the other important work as well, especially, letting some of our flavivirus people think about dengue as well, instead of, okay, well, you're here and we trust your hands and you've kind of passed all the screening steps to be able to start training into the BL3. So you're not going to do dengue now. We're going to make you a BL3 person.
One of the things we always said in the lab was the first experiment is to clone yourself. And then
I would love that.
Your clone can do half of the experiments. Yeah.
I think it's really important to do the basic science, because we're at this point because there wasn't enough basic science with coronaviruses before, and people are not recognizing this enough, that it's not just making vaccines and antivirals. You need to know fundamental stuff that we don't have. And so you have to do that. It's really important to emphasize. I also want to say that we've done a lot of your papers already here on TWiV, because you're on most of them. So we have had a good time looking at that. All right. Our guest today from the University of North Carolina, Chapel Hill, Lisa Gralinski. Thanks for joining us.
Thank you so much.
And good luck.
Thanks, Lisa. This is great.
Good luck going forward. Really stay safe, and we look forward to collaborative cross paper.
Yes.
Thanks, Lisa. This was awesome.
Bye-bye.
Thank you, everyone.
Back then. Yeah. Bye.
Well, that was great. I really enjoyed that.
Fun.
Yeah, I learned a lot.
And Vincent, wonderful selection of papers to talk about.
Yeah, we had talked a lot about what she's published on 2 already, so I thought we could go back and look at some of these founding. Everything good?
Of course, I forget everything we've talked about.
Throughout the months of October and November, all donations made to Parasites Without Borders will be matched and doubled and donated to Microbe TV. Go over there, parasiteswithoutborders.com. Amy Rosenfeld here at Columbia still has a lab position available to work on plus-strand RNA viruses that lead to respiratory and neurological diseases. Applicants should have a BS in science, some lab experience that is not a lab class. Email tech@microbe.tv. And the Biological Safety Program at Columbia has an opening for a biological safety officer. In the age of COVID-19, biological safety professionals have never been more needed, valued, or felt more purpose in their calling. The ideal candidate would be someone with a life sciences degree, bachelor's or higher, at least 2 years of lab experience, who is hungry to learn about infectious agents and molecular biology. More information and application instructions are available. We'll put the link in the show notes. Whether the show notes, you say microbe.tv/TWiV. We have a paper that Kathy brought to our attention earlier this week.
Right.
So it's your fault, Kathy.
Yeah, right. Well, no, it came from one of my friends from graduate school who has become a TWiV listener. And she heard this author present the work in their journal club. And the thing that triggered them was that they had read some paper in the late spring about a receptor that they work on that's was a potential or is a potential SARS-CoV-2 receptor, and they've worked on it from the pain relief standpoint. And the paper mentioned that spike binds to this receptor as in addition to binding to ACE2. And so this investigator's name is Rajesh Khanna. And he said that in the journal club that there has been some anecdotal evidence about people who suffer from chronic pain who feel great in pre-symptomatic days just before they get COVID. So he wondered what was going on and tested the hypothesis that this pathway that his lab knows so well is activated and somehow is relieving or masking pain. And so they got everybody in the lab organized and did these experiments in rats. And so the title of the paper is SARS-CoV-2 Spike Protein Co-ops VEGF-A Neuropilin-1 Receptor Signaling to Induce Analgesia. And we first became aware of it in bioRxiv, but it's now published in Pain, another one of those one-word journal titles, which is open access. But when you go to that, it still is the double-spaced manuscript form of the paper. So it's not been PDF formatted yet.
This one, however, can be highlighted and saved in its highlighted form.
To its credit.
Someone offered to send me, this is funny, complaining about not being able to highlight and save PDFs. He offered to send me a disk. He said, I have a version of Acrobat, which is government only because they're not allowed to save whatever. And he said that this will work, but I'm afraid of him to send it to me because I don't know what it will do to my computer. Isn't that weird? Anyway, yeah, so this is interesting because it's different, and this neuropilin paper has just come out suggesting it's involved in entry of the virus into cells, and maybe we'll do that on Friday. So neuropilin receptor, the spike domain, SARS-CoV-2 spike can bind to it quite clearly. And interestingly, in COVID-19 patients, there is upregulation of neuropilin-1 receptor. And so they asked here whether spike, so there is a natural ligand of neuropilin, which is VEGF-A and neuropilin-1, and they asked whether spike could interfere with that. And analgesia, of course, is taking away pain, right? Mm-hmm. When you take aspirin, and aspirin is an analgesic. So who would think that spike would be an analgesic, right? But who knows? So as Kathy said, they do a variety of experiments in rats, both living rats and also in cells, neurons from them. And they do electrophysiological experiments looking at conductance. And I thought as I was reading this really should have been done on TWiN because Ori Lieberman is an expert on these kinds of conductance experiments. But I put it on the show notes before I had read it. So here we are. And we will bumble through it. But
Yeah.
So they basically say they confirm this interaction between spike and neuropilin-1, is it neuropilin or neuropilin-1 is the ligand or the receptor? Now, I'm going to mess this up already. Or NRP1, is that the cell membrane protein that's involved in entry?
I'm pretty sure it's the receptor.
I think it's the receptor.
Yeah.
And the
It's,
ligand
they've got
for
this
that.
diagram in the first figure down in D,
Right.
Okay.
that identifies all the different molecules. So NRP1 is a receptor, yes.
Okay. And VEGF is a natural
VEGF-B,
ligand.
VEGF-A is a ligand.
Yeah, and there's some other ligands as well. And then there are natural inhibitors, drugs, small molecule drugs that can inhibit. All right, so they confirm the interaction of the spike with NRP1 by ELISA. All right, they put neurons, sensory neurons on plates, which they can get from rats, and then they can measure nerve firing in these neuron cultures. And so when you add VEGF, it causes firing of the nerves, and it's binding this NRP1. And if you add spike, it blocks this firing. Okay? And I'm saying firing, but obviously it's a extracellular action potential, which they can measure electrically. And these, both VEGF-A and spike have a binding pocket on NRP1. So then they do an experiment. So then they say, okay, this in cell culture, spike can block VEGF-A firing. What happens in an animal? So they have a paw withdrawal threshold assay. And I think basically they're putting a needle intraplantar, which means between the toes, right? And seeing how quickly the rat pulls away from it. So the pain threshold, how much can they take? Paw withdrawal threshold. It's very hard for me to say those words. Too many W's. Okay.
We can tell you're from New Jersey.
Really? Well, you live there, man. Hey, you're going to get contaminated too. You'll see.
I know.
If you live there. Hey, Rich, can you say that? Paw withdrawal?
Paw withdrawal.
You got a southern accent.
Yeah, but, okay, so I'm doing a sort of a Baltimore slur through the last syllables of withdrawal.
Okay.
Right?
Yeah.
Okay, so this is a tough sentence. VEGF-A is proneoceptive, as intraplantar injection of VEGF-A decreased both paw withdrawal thresholds and latencies to a thermal stimulus. They can also put some heat on the paw and see how long it takes for the rat to withdraw. You know, if you have analgesic, you're going to withdraw later. And then if they put spike, it blunts the pain and the thermal hyperalgesia induced by VEGF-A alone. So VEGF-A is, increasing the pain response, and spike is blunting it by binding to NRP1 presumably and interfering in some way, I suppose. Is that all correct? Does that make sense, folks?
That sounds right to me.
Mm-hmm. Then they say, what's the mechanism? So they look at ion channels, and there are all kinds of ion channels that are involved in neurotransmission. They look at sodium channels, where they know that VEGF-A increases sodium currents, and in fact, spike blocks it. So spike blocks the increase in sodium flow through channels that's stimulated by VEGF-A. So that's mechanistic explanation for what's going on in the pain experiments. They also look at calcium channels, which are important too for neurotransmission, especially in pain-related transmission. VEGF-A, again, increases calcium currents, and it's blocked by spike. So spike blocks both sodium and calcium currents that are stimulated by VEGF-A. What else did we? They also looked at synaptic activity measured electrically, and this is in neurons. And VEGF increases synaptic activity, and it's reduced by spike as well as a small drug inhibitor of that. And they say it's...
Yeah, pretty much through the whole paper, they do this other inhibitor and spike kind of, I think, as a pharmaceutical control.
Right. And then the last experiment is, it's called the spared nerve injury model of chronic neuropathic pain. Okay, to evaluate the potential of disrupting the pathway with spike. So they're basically in rats, if they give spike intrathecally, right, that's right into the brain, right, it decreases the phosphorylation of the target, the downstream target of VEGF-A, basically, in the nervous system. So in rats, spike can inhibit signaling that is part of this pathway of chronic neuropathic pain. Its spinal administration of spike decreases pain as well. So basically, the finding is that spike alone can bind this receptor, interferes with signaling, and can dampen pain responses. And it's the VEGF-A NRP1 pathway. So they say spike protein is analgesic in this nerve injury rat model. Now, I cannot properly evaluate the electrical experiments, but the only thing I worry about is how much spike they're using and whether that is level that you would see in a virus infection, right? And there are two things. You can have virus particles with spike on them binding to NRP1. And then there's soluble spike, which is shed from virus particles. And we know that happens. We know that spike can be shed, right? I just wonder how much soluble spike there is around, whether it gets into the CNS so it could do this. I would actually have liked to see them do these experiments with virus, but of course that's a BSL-3 experiment and harder to do. But maybe spike on virus can't do this for some reason. I don't know.
Those were exactly the experiments I was wondering about, both the spike on virus versus free and the experiments in rats. And I specifically wondered whether there were differences with different types of neurons. And that's about, where my neuroscience background ends. But I could imagine that if you were to infect rats with virus, only some neurons would be in contact with spike.
Yeah, that's right. It's not.
And so would the particular types of neurons that had this effect matter?
Yeah, so I think in the end, because spike is binding NRP1, you see these effects. Whether this is happening in infected people or not, I don't think is answered by this. And maybe if this paper generates enough interest, people will do that, right?
Could they potentially look at things they're seeing in some of the spike-only vaccines to see if they see any strange effects with pain in any of the people?
That's a really interesting
I
question.
suppose so. Yeah. These are like the mRNA spike-based vaccines you're thinking about, or the vectored ones even?
I mean, in theory, I'm thinking, a subunit is the most obvious, but any of the spike vaccines, potentially, maybe patients could be reporting pain-related changes.
Right. It could be that it was just coincidental and they put the pieces together and they found this information. Or it could be that there is an effect of the virus that's reducing this chronic pain just before the infection becomes full blown. They do say in the discussion, leveraging this atypical function of SARS-CoV-2 spike protein may yield a novel class of therapeutics for pain, which is their interest. In fact, in the end of the manuscript where they talk about potential conflicts, now I can't find it, but they have, the PI has some patents and interest in, here it is, a company developing non-opioid drugs for chronic pain. So they're really not interested in the virology per se, but the pain aspect.
But these little drugs they use that are positive controls, I guess those are analgesics already, right? So I don't know how spike would improve on those. And anyway, you wouldn't want to use
different shapes of molecules and maybe things that you would use in combination, or
But you wouldn't use spike. it's
No.
too big.
And I think they're even just saying that it's just a portion of the spike.
Yeah.
And, you could, how
You could mimic it,
pharmacy guys change the molecules and make them better and more bioavailable and PK and all that kind of stuff.
Maybe there's a peptide you could use.
Peptide or a small molecule you could design. I think it's interesting. Anyway, it's something different. That
right.
That's why I thought it was of TWiVable.
Yeah, and I'm not sure that we mentioned that these investigators are from the University of Arizona, and the first author is Abin Mutal, and there are a bunch of other authors, and Rajesh Khanna is the senior author.
You don't know, what did you say? Do you know some of these people, or you, Kathy?
No, a friend of mine went to their journal club that Rajesh Khanna presented this work in.
Got it. Okay.
So that's how we got a little of the backstory.
Well, I think if you read the journal Pain, you will have some pain because I can't understand everything.
Or at least we will have pain. These assays in here were.
This was a struggle for me. This is beyond the boundaries of outside my wheelhouse.
Sorry,
outside my expertise.
I think twin guys would have done it better, but I think you get the point, right? You get the general point. Sure. There's some good evidence in these culture and animal models that this is inhibiting a pathway that leads to pain sensation. So. And Kathy notes this is a one-word journal. There are quite a few, right? We have cell and neuron.
Virology,
science, immunity, nature. Yeah, there are a lot, right?
Yeah.
There are a lot. However, to take it one step further, there are not a lot of 4-word web domains available anymore. If you want to buy them, like TWiV, you could never get, well, because I have TWiV, but 4-word web domains are the fewer the letters, not 4-word letters, 4-letter web domains, 3, 2. Does anyone have a.com? Let's see. No, there's no a.com.
If you get the GoDaddy ad saying you could buy this domain, then you'll know.
So there's no a. There's ab.com. Yeah. It's probably going to be a horrid site. Allen Bradley products. Yeah. So the few letters are hard to get. And then when I got TWiV, someone said, "Hey, it's pretty good. You got TWiV." I didn't think much about that anyway. Let's do some email. I will take this first one. This is from Brigitte. Hello from the Laurentides, 1 hour plus north of Montreal, where fall and most leaves have already fallen. Wow. We still have all leaves here. They're just starting to turn, with nights hovering around 0, 1, 2 Celsius and days creeping a little below 10. I'm pretty much newbie to your podcast. I learned about it this summer thanks to one of my favorite authors, Malcolm Gladwell. He talks about it in one of his podcasts. You're a newbie, but you get the weather, don't you? Little did I know that I would get so seriously addicted to your talks. I want to thank you so much for getting science back into my life. I'm not certain if one of you speaks French, but I forward you a note below. I just sent to the Montreal newspaper La Presse, as I read an article in it this week stating that a local survey showed 84% of the population have confidence in science and scientists. Obviously, you will find this as great news. That's why I suggested they should have an article in their newspaper about TWiV to inform people about what to do in your podcast. Then thinking further, I also suggested that they might want to include a weekly summary of what your latest podcast talks about as a teaser, i.e. who were your guests, what papers you reviewed, etc. I was wondering if this is being done in newspapers anywhere else. Maybe another way to reach more, quote, regular, unquote, people. Hey, we're regular. Increasing their understanding and knowledge of what scientists do. Anyways, just a suggestion. Love your passion. Keep being yourselves. Vous êtes magnifique. Je vous adore. Cheers, Brigitte.
That's excellent French.
Very nice. French was the second language that I learned. I had proper schooling. I took 5 years of French in high school and college. So I do remember, but I took Italian. I learned Italian growing up. My family And so now I mix up Italian and French when I try and speak one or the other.
Well, French is all I've got, and that sounded good to me.
So she sent her letter, which I Google translated. Vous devriez jeter un coup d'œil à un podcast de science que vous trouverez sur Microbe TV, etc. You know, you should take a look at this podcast. And she gives, listen to TWiV with Fauci and others. And I'm thinking you might want to do a synopsis of the discussions in every week. So this is a great idea. They
ought to have a little box in the upper right-hand corner of the front page of the Washington Post and the New York Times that summarizes TWiV.
But they won't do it. They won't do it. It's too much work. Many people have said we should do it, but it's a lot of work to do that. And not that I mind work. It's just there's not a lot of time for it. If anyone would like to make it, Kiki was doing summaries, but
well, the little summaries that you even write that get posted with the podcasts.
Yeah.
That kind of says what we talked about and who the guests were.
Those are not easy to write, by the way. Sometimes I struggle with them. And they go through multiple versions, so it gets clear, right? Yeah. And I always want it to be, for some reason that I don't understand, I want it to be one sentence, right? I just think it's less intimidating. And so getting everything in one sentence is challenging, but I do it. Kathy, can you take the next one?
Jessa writes, "Hi, Vincent. I listened all the way to the end of episode 669, Raspberry Blow. I'm a COVID ICU nurse in San Antonio, Texas. Am I your lone Texas fan? LOL. I have been listening to your podcast all the way to the end for the past few weeks. Thank you and all of the other scientists and doctors on the show for your up-to-date information. It helps me in my daily care of these patients. Have a great day. Regards, Jessa."
We have a bunch.
Texas is a strong TWiV fan base.
Yeah.
As a matter of fact, I was thinking the other day that, when we're all out of the pandemic, we ought to have an Austin metro area TWiV party. There's enough listeners around here so that we could all go out to lunch together and have a pretty good time.
Maybe have to meet in Buda.
Absolutely. That ought to be done. Yeah.
Rich, can you take the next one?
Right. Joyce writes, "Dear TWiVers, this is a note to first let you know that, 1, I'm from Texas. Hi, Rich. And 2, yes, I listened to your podcast all the way to the end." This is a listen to the end arc, Vincent?
Yep.
That's because I don't want to miss a thing, Dr. Racaniello. My chemical engineer husband, teenage daughter, and I, trained as just an ecologist, have all had your show in our podcast players since we first discovered it this spring. We are so grateful for your friendly voices. It's like Journal Club, but better. You say the show is edutainment, but for my family, it's more like edu-comfort. You remind us that there are a lot of good people out there doing good work, and we can support them in their efforts. All is not lost after all, at least not yet. With fires to our west, hurricanes to our east, and great financial need all around, my family has tried to pitch in wherever possible these past few months. In early August, my husband created a national country-level web app to allow people to visualize what kind of exposure risk they might face when they move throughout the community. She gives a link to the app. In parentheses, the page settings include the methodology. Of course, the app is only as good as the data feeding it, and we're finding in Texas that, as Rich has mentioned, our county-level data can be hit or miss. Perhaps it's more reliable where you are. In any case, as areas continue to open up, we hope people understand at least somewhat the potential risks, even if they have not yet been personally touched by COVID-19, and continue to do all the things that we know work: masks, hygiene, distance, and lots of fresh air. Finally, to help mark this unusual year, I'm hoping for some TWiV merchandise in my Christmas stocking, and will be dropping the appropriate hints. May I make a comment about the t-shirts, however? women must sometimes be a little more careful about the placement of logos and designs on shirts. The miasma of politics tees may be a little more flattering with those phrases on the back or if on the front with the TWiV logo very small underneath. If you'd like help making some adjustments to your graphics and layouts to allow more flattering options, let me know and I'd be happy to assist. I'm not a graphics person, but do know my way around some graphics tools as a parent volunteer and do like to wear nice t-shirts that advertise my favorite causes. Gratitude to you all, Jay. Well, that's interesting. That's one I hadn't thought about. Good point.
So that's a cool app that you might want to check out, Rich.
Yeah, I will. I have not checked it out until now, but I will.
The odds location map app, yeah, because I went in and you can put in your specific state, and I put in Washtenaw County, and it gives the probability of a COVID-19 positive individual present at a restaurant with 80 people, or a party with 30 people, or lunch with 4 people, and so on. It's good.
Very interesting. So I have to say the county by county reporting in Texas is improving with time. These things get better if there are appropriate people in charge.
And I sent Joyce. I sent Joyce the graphics files for the T-shirts, and she said she'd work on them. So, because I don't.
Some of these numbers for Morris County are scary.
That's where you are?
Yes.
Well, the numbers are way up all over, right? We're up 36% over the last 2 weeks in the U.S. 60,598 cases yesterday. Man, I mean, now that's the third peak. It's going up. It's quite clear. It's not going away, folks. So despite someone saying that or many people saying that, it's not going away. Get real.
So I had an email from one of my local colleagues this morning asking why, with respect, in particular, since we're getting resurgence almost globally, why China is so quiet relative to caseload, whether we're getting all the news or whether they really have it strictly under control. And, I couldn't really answer that except that from what I know, at least in urban areas, if there's anything that happens, they step on it with really stringent, Tetris right away. Yeah. But I've always wondered about circulation in the rural areas in China and, what actually is going on. I don't have an answer there, or whether it's just a reporting issue. I don't know.
We have lots of listeners in China. Maybe some of them could chime in and let us know what's going on.
That would be good, actually.
That would be really helpful.
I wonder, what is the travel situation to China? Is travel still limited? Does anyone know? I don't know. I don't know.
I mean, travel for people with U.S. passports is limited to most countries.
Technically, Brianne, people are not supposed to travel from New Jersey to New York anymore.
I know. I saw that.
Because we have now enough cases in New Jersey that it goes over the limit that New York says you should have before you come. But Cuomo's not doing it because so many people work in New York and live in New Jersey that.
Yeah, look at you in your office today.
Yeah, I miss what they said. If you're essential, I'm not essential by any means. I am non-essential, but I am here.
So I've made a comparison of Morris County with Washtenaw County, and Morris County, New Jersey, 39 average new known cases per day as of October 18th. Washtenaw, instead of 39, 74.
Wow.
The university and the county announced yesterday that undergraduates are limited to remaining in Washtenaw County for the next, until election day, in fact, till November 3rd. If they want to leave campus and go home, they have to go through a regimen of testing and so forth so that it's clear that they're not taking the virus home to their community there. It was not a response that I was expecting to have the county jump in and then limit them to the county. So
yeah. The good
I'm going
news.
to bookmark this app and play with it. This is quite something.
yeah.
The good news is that here in New York City, the schools have had fewer cases than they expected. So they seem to be doing something right.
I have the sense, this is just a sense, I don't have any data, but I have the sense that masking and distancing and hygiene are even more powerful than we have suspected in the past, and applied religiously can do a lot more good than we might have even anticipated.
Yes.
I have another comment on this letter, because she talks about what the podcast means to her family and edu-comfort. Okay. This is beyond just the science. I had an opportunity to be on another podcast yesterday that was a little more overtly touchy-feely than what we ordinarily do. And one of the questions was, knowing that I was involved in this, what my motivation is. And, of course I said, I really like doing this. I really like discussing the science. But what I missed the opportunity to say is that I just like hanging out with you guys. I've always, envied people who had, I thought when I left work, I was going to miss the society of work, hanging around in my office and having conversations. I have friends who have, a daily or a weekly sort of coffee clutch where they go to some sort of restaurant and hang out with their friends. And, aside from work, I've never had that sort of social circle, but here I do. And this is a significant part of the motivation, just hanging out with you guys. It's great.
Yeah, it's fun.
So thanks.
We started it for
Thank you.
We started it for science, but in the end, you end up talking with your friends. Yeah.
Yeah. I think the part that I look forward to as much as anything is the pre-show conversation and the post-show conversation.
That's right.
As well as the science during the show. So yeah, I agree.
Brianne, can you take the next one?
Sure. Graciella writes, "Good morning, TWiV team. This is just a feel-good gratitude letter, since I am tickled pink with some of the wittiness from your show. I thoroughly enjoy listening and am learning so much. I look forward to new episodes, and though it is more convenient to listen, I find myself logging on from my desktop so I can actually watch you guys with all your personalities showing through the screen. Thank you for all the time you put into this. You are appreciated. And this gal from Texas does listen to the very end. Gratefully yours, Graciella."
I think I purposely put 3 Texas letters in a row, right? And they all listen to the end. But it seems that only Texans listen to the end, right? I know other people do. Wayne writes, "Bozeman is in Gallatin County. COVID is spreading rapidly in Montana." And he sends a map. Now, we have heard previously that Montana was largely spared, including Bozeman, and now that's no longer the case. And he sends a link to an article on NBCMontana.com about the outbreak. 600 some cases. Numbers are going up. I fear it's going to get worse this winter. I think you're probably right. No place is going to be spared. Wherever there are people, this will be going. Here in Wakayama, which is in Japan, so Wayne lives in Japan, we remain at having 238 folks infected, no new infections for weeks, and only 4 deaths early on in this pandemic. Very good. Masks work. Personal distance works. Taking this disease seriously works. Rural Japanese get it. Young urban Japanese in cities like Tokyo and Osaka don't, and the numbers show it. That's great. That's just what you said, Rich. Masks.
Yeah, this is maybe the answer to the China question, too.
Taking the disease seriously. That's so many people still in the U.S. do not take it seriously. And I do not understand why it's politically motivated not to take it seriously. I hear people on certain talk shows saying it's nothing to it. I don't get it. It's not about politics. It's about science, folks. And then Wayne gives us a haiku, I guess. Is this a haiku?
No, too many syllables.
What would you call it? Just a poem, right?
Mm-hmm.
Yeah.
A tsunami of virus washes over the land, clinging to scientific truth. Some are saved, others drown, holding on to rocks of ignorance. I love it.
Mm-hmm.
Rocks
Beautiful.
of ignorance.
And since you gave us the information about Gallatin and Bozeman, I looked it up. It's between Morris County and Washtenaw County in terms of number of cases. It's 47. That's a cool app.
It is really nice.
I have a sense too of the sort of changing dynamics of this. It's like, in my mind, it took, 6 months or so for the virus to actually spread globally. Okay. But now we have a foundation of virus everywhere.
Everywhere.
So that, any lapse in vigilance and it pops up right there.
Oh, you can bet.
It's already there.
You can bet the virus is still in China. It is still in China, and it will pop up if they relax. Yeah, for sure. It's not going away. Kathy, can you take the next one?
Uh-oh.
What's
let's
wrong?
See. Well, I was looking
She's been
at
cruising around.
the link
Right.
down
It's
below
okay.
the
She's
NBC
lost
link.
in internet space.
We're not gonna. We're not gonna worry.
Martin writes, "Dear Vincent, I've just spotted this number plate on my TWiV walk. Maybe the owner is a fan of ASV too, and it's a car with a license plate BU65 ASV. Best wishes to the team, Martin." That's cool.
It's not U.S. It's somewhere else, right?
Right. Yeah.
TWiV walk.
So we're in That's cool. That's very cool. Rich.
Paul writes, "Hi, Vincent, and all the wonderful guests you have on your shows. I live in Southport, England, near Royal Birkdale Golf Course, and I've been following TWiV for some time. Recently, I saw an episode where you discussed free healthcare in the U.S. and other places. I've spent the last 12 months working for a U.S.-Canada healthcare company in a royal hospital in the UK, so I'm very aware of my colleagues' reliance on the company insurance policy in the US. I wanted to just be a bit pedantic. I know you like that, about the wording. In the episode in question, it was referred to as, in quotes, free healthcare. It is not free. Wherever it is given, someone has to pay for it. It is really called free healthcare at point of use. In the UK, we have the National Health Service, which is a fantastic Labour Party created system. Everyone who works in the UK and earns over a certain threshold pays national insurance contributions, which funds pensions and the national health system with general tax income from the government. Sadly, funding of the NHS under conservative governments has fallen to the lowest in 50 years up until the coronavirus pandemic. We also have private hospitals, of course, and many employers offer private health insurance as a perk, but it is usually only for outpatient treatment in quieter hospitals. Often the work you have done is by the NHS doctors and surgeons anyway, and gives a link to a wiki article on the National Health Service in England. All in all, everyone who is a resident or citizen is covered for free at the point of use, and those who can pay for quicker treatment, etc., can still do so via private medical routes. If you are an overseas visitor and need hospital treatment, you will have to pay later, but the cost will be considerably lower than in the US, for example. There are plenty of YouTube videos about this. Anyhow, you guys really make me smile and fill my head with great info. Of course, as a 50-year-old, keeping the new knowledge in my head is another matter. And gives this, as an example of that, gives the same link again. Okay. Kind regards. Kind regards, Paul. Those are important distinctions. There was a series, a 4-part series on the PBS NewsHour recently where one of the investigators went around and investigated healthcare systems in 4 different countries, the UK and Switzerland, and I forget the other 2. And it was very interesting, the different approaches to this. And I anticipate, I hope, that we're going to go through another round of how do we do this in the U.S. Within the next few years. Matter of fact, I fully anticipate that if the Supreme Court winds up trashing the Affordable Care Act in some fashion or another, the outcome will be to motivate the U.S. Congress to come up with an appropriate replacement to take care of the problem in even a better fashion than the ACA. That's my optimistic spin on things.
But he's right. It is paid for at some point. It's true.
Yeah, somebody pays for it. As a matter of fact, it occurs to me in the U.S. that people complain that they don't want to, pay for services that other people who can't pay for it don't get. But the fact is that to a very large extent you are. Hospitals have an indigent care budget.
Yeah.
Okay. Because, doctors, hospitals, they aren't going to let people die because they can't take care of them. People come in, they can't pay for it. They take care of them anyway. And they have a certain discretionary budget that is precisely for that. Who pays for that? The other customers, of course. Okay.
Yeah, my go-to example for that is we all pay for the head injuries of people that don't wear motorcycle helmets.
Yep.
And smoking injuries, right? Lung injuries, sure. Yep, absolutely. People who don't wear masks. All right, one more, Brianne.
All right. Richard writes, but not this Richard. Oh. Dear Vincent and team, I have been listening from the start of the pandemic and have found the show incredibly informative and interesting. Many thanks for your efforts. After discussing some of the show content with my mom, we wondered if there was a possibility for people to asymptomatically carry the virus at low levels for a significant period of time, and therefore cause infections like typhoid Mary. Indeed, I was also wondering where the common cold coronaviruses go in the summer. Do they just circulate at very low levels, or are there people who are reservoirs for these, and once their noses start running in the winter, we see the typical winter resurgence? If this is a possibility, then could places like New Zealand suffer random unknown outbreaks due to carriers of this sort? Many thanks, Richard. Those are good questions.
which is the scientist's way of saying, I don't know.
Well,
I would guess that the common cold coronaviruses circulate at very low levels.
Yeah, they do.
I think
That's right. No, all common cold viruses. In fact, there was no corona in that question. It was just common cold viruses. All of them. They're always at low levels. Say here in the temperate zones in the summer. People get flu throughout the summer. It's just not reported, but it's there, and that's what keeps it there for sure. But I'm not aware of any evidence that you have long-term infections because most are over in 2 weeks. Most of the shedding is over. If you're very sick, it's extended a bit longer, but eventually, as far as I know, who knows? Maybe we'll find out. In the rag mice that Lisa was talking about with no immune system, she said virus, I guess that's SARS-1, right? They persist for long periods of time. So it's always possible we might find it in humans. But right now, there's no evidence that these SARS-CoV-2 causes months and months and months of infection and shedding of infectious virus.
This, however, gets into questions of basic virology and patterns of pathogenesis that are good questions because there are viruses that can hang around in an individual asymptomatically for extended periods of time. Hepatitis B hepatitis C.
Sure. Yeah.
Other viruses as well. You can have chronic carriers who are asymptomatic and spread virus. So we call those chronic or persistent infections. There are also certain latent infections where the virus can basically hide out and not cause a problem, but occasionally reemerge. Herpes virus infections of one sort or another, HIV. So there are different, not all viruses behave the same, and there are whole families that have different patterns of pathogenesis. These coronaviruses and others fall into a pattern that we call acute, okay, which is basically hit and run. You get infected, you cure the virus infection and move on. And I don't know any evidence in these that there is a lingering persistence. However, we don't know everything yet.
Okay. Let's do some picks. Brianne, what have you got for us today?
So I have a science podcast that I recently came across known as The Lonely Pipette. They have 4 episodes, although one is episode 0. So the most recent episode is episode 3. And they have Harmeet Malik. I'm just noticing that I spelled that wrong in the show notes. He talks a lot about sort of science and science mentoring. And it's a really lovely episode and a lovely discussion with Harmeet. And I think a lot of our listeners would enjoy.
Harmeet is one of my favorites. I'll have a listen to this. Very good.
Kathy, what do you have?
And I promise I do know how to spell.
You could chalk it up to auto-correcting.
That's probably it.
So I have 2 picks. The first one I'll mention is the Wear a Mask parody of Be My Guest, which if you haven't seen it yet, I mean, it's really been going around virally. It's very, very well done. And it's about 2 minutes long. So we have the link for that. And then the other one is Dr. Boyd the Chemist. Mm-hmm. And I came across this because he was featured in the daily newsletter that we get online. And so he's a University of Michigan graduate. He works at the Naval Research Lab, and he has produced a whole lot of really great short videos. The first one that roped me in was one that was linked to from the Michigan article about what's an experiment versus a demonstration and what is a hypothesis. And then I was down the rabbit hole. I've just listed several of the favorites here. There's one about oobleck, how to read an old school thermometer. And then he does one about where would vibranium go on the periodic table. So vibranium was from the Marvel comics. And then at the end, he kind of raps about a new Marvel storyline. And it's really worth hearing that. And then it looks like he's pulled that out and made a couple of separate videos about vibranium and Marvel and so forth. And they're usually around 4 or 5 minutes long. He's wearing his lab coat. He puts on his glasses. If he's doing something that's going to make a mess, there's always a little safety warning, but nothing's ever really dangerous. And it's just great. I think, kids who want to be introduced to science or who like science would enjoy this. And then older kids like me who just like these kinds of experiments and fun can also enjoy it.
Nice.
I am really excited to watch this vibranium video.
Yeah.
Yeah. Kathy sent me these links earlier and I've had a look at them. They're quite good.
Yeah.
Rich, what do you have for us?
So I'm taking a bit of a flyer here because in contrast to my standing pick policy, I have not listened carefully to what I'm picking here. Okay. But I trust the source. This is a podcast called Clear and Vivid with Alan Alda, who, if you'll recall from a previous letter, sounds like Vincent Racaniello. But at any rate, Alan has a passion for science communication and in fact runs an institute that we've picked in the past, or a program at State University of New York in Stony Brook that helps scientists learn how to communicate science to the lay public. And this is a podcast that's sort of a sidelight to that, where he interviews a bunch of different people, not necessarily all scientists, as a sort of a vehicle for communicating science and other concepts to the lay public. So I've just had a look at some of these. I'm listening to one now where he interviews Neil deGrasse Tyson. And so I look forward to putting this in my podcast stream.
Hey, maybe we should get him on TWiV.
Actually, that has been suggested, and we should do that.
Would have no idea.
I think we maybe have enough cool at this point so that he would like to visit us.
I noticed that one of his guests is Leslie Vosshall, who is a mosquito biologist at Rockefeller. And we tried to get her on TWiV a number of years ago, and she couldn't be bothered. She said, I'm too busy. Get one of my postdocs. But when Alan Alda calls, boom. So thank you, Leslie. All right.
Well, maybe you have to drop Tony Fauci's name. He's been on TWiV twice. Wouldn't you like to come on?
I use that now with some people. So I use that with Ed Young. He's coming on TWiV in a couple of weeks.
Oh, good.
Well,
I dropped the Fauci, the F-bomb, as they say.
Yeah, I think trying to get Alan Alda on makes a lot of sense. I think he'd enjoy it.
Mm-hmm.
Yeah, he would. I would use the Tony Fauci bomb for sure.
Yeah.
All right, so we have 3 podcast picks this week. Mine is Immune number 36, which we just recorded yesterday, which I think is really important. It covers a paper that sheds a lot of light on the aberrant antibody responses in severe COVID-19 patients. Basically, the lymph node germinal centers appear to be trashed very early in infection. You don't make good antibodies. You make antibodies outside of the germinal centers, which are not highly mutated. They're low affinity, and there's no memory. And this is probably because of the cytokines that are being produced. So really interesting paper. We had a guest, Alex Ward from University of Indiana School of Medicine.
I think it was Alex Dent.
Dent?
I think it was Dent.
Well, I got 4 letters, right? Yeah, it's Alex Dent. I'm sorry, Alex. But Alex won't know because he will never listen to this. Alex Dent, yeah. I have problems even at 67. This guy has problems at 50 remembering. Wow, that's pretty young. Anyway, check that out. We have a listener
You remember all of the parts about the germinal centers and things like that?
I remember the important stuff, right? Yeah, the science.
And you should remember Arthur Dent from Hitchhiker's Guide to the Galaxy.
Yeah.
He's a fictional character, though.
Yeah. Anyway, check it out. Immune number 36. And we have a listener pick from Ryan, which I think I had on the list Friday, and I forgot to mention it. It's a YouTube video where Dr. Fauci appears on CNBC to talk about COVID-19 and the politics surrounding the pandemic, and how politics have played a big role this time. Although I think probably in every pandemic they do to a certain extent, but heightened this time around. All right, that is TWiV 674, microbe.tv/TWiV for show notes. Send your questions and comments to TWiV@microbe.tv. When I put together the emails for these shows, I love it because you all write such great things. Even if you're critical, it's just well-reasoned and thought out for the most part. I really enjoy it, so keep it up. TWiV@microbe.tv. If you like what we do, consider supporting us. Now, of course, ParatesWithoutBorders.com is doing the matching donations. So if you want to give some money, that's a good way to do it until the end of November. But if you want to do it after that, microbe.tv/contribute. And of course, if you buy stuff on Amazon, use our affiliate link because we get a little money from that and it doesn't cost you anything extra. Brianne Barker's over at Drew University on Twitter, BioProf Barker. Thanks, Brianne.
Thanks. It was great to be here.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
And I am Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.